Okay, how's that?
Perfect.
We all on the PowerPoint? Perfect. All right, so thanks everybody for tuning in. Thanks, Janae, for the introduction. I'm going to be speaking to you a little bit tonight about, well, could be this morning, depending on wherever you are in the world, but about some of our manta ray research in Mozambique. And I'm going to focus on 3 different aspects of that research: population trends, movement patterns, and also population genetics. And these are sort of 3 of the many research techniques we use to learn about these incredible animals, and that information goes towards guiding more effective conservation and management. And I'm just going to start off by introducing myself. My name is Steph Venables. I have been working with manta rays since 2013. I actually started off working in the northwest of Western Australia at the Ningaloo Reef. And I did my honours research up in a place called Coral Bay, where I was looking at tourism interactions and their impacts on the manta ray population there. So that was a very eye-opening year for me, my first year out of undergrad. I thought I was going to be studying coral reefs and coral restoration, and I ended up studying these amazing giant rays, and from that moment I was hooked. So I began working with MMF in 2014 in Mozambique, where I was a research assistant for around 6 or 7 months, and I was running the Bazaruto Satellite Research Station in the north of the Inhambane Province. And then since then I've been doing a bit of work in Raja Ampat in Indonesia, so co-managing our manta ray project out there. And in 2016, I thought it was a good idea. And now that I'm looking back, it was a great idea to start a PhD with the work that I've been doing with MMF. And I've put almost doctor at the bottom of this screen here because I've just received my thesis report and I passed with minor corrections. So once I get those corrections in, I will be Dr. Steph. And I wanted to start this webinar off just to give a little idea of what even is a PhD anyway. I know that a lot of people in my life have got to know what I mean by a PhD and what I mean by a thesis, but I thought I would just explain it to those who might not be surrounded by academia, and just to give you guys an idea of what I say when I mean thesis. Now, it can vary in different countries, different institutions, and different fields as well. So this is just an example of an Australian thesis, and it's all put together in the end into one document, into which grad students pour the blood, sweat, tears, and their soul into. And it usually starts off with a general introduction, which gives you an idea of what's known so far about their research topic. And I've put in brackets a literature review, because a lot of people will actually do a more in-depth review of all the scientific literature about their subject that is already known. That's quite common when you're looking at something new or you're looking at a new sort of technique. I didn't do one of these, which is why it's in brackets. And then you have multiple data chapters which follow. And so these, I guess, are the heart and soul of the thesis. I think our minimum at the university that I was at was 3, but it can range. I know people who have 8 or 9 data chapters, and each is structured sort of as a separate scientific study, and they're written more commonly nowadays into publications. So each chapter can be a standalone scientific publication, and we try and publish the studies as we go along. It's good practice, and it's also a way of getting your science out into the world as you go. And then it's all wrapped up together into a general discussion which sort of discusses future research possibilities. For me, I talked a lot about conservation and management implications, and it sort of draws that all together. And I've put in the little picture at the bottom because it's one of those things, and 2020 has taught everyone this, is that your plan of how you think your thesis and your PhD is going to go is never really what it seems. And so each individual student has their own sort of challenges along the way. So if you do know anyone that is doing a PhD, just go and give them a hug if that's socially acceptable at the moment, and tell them that they're doing a good job. All right, so on to the good stuff. I'm going to be speaking a lot about Mozambique in this talk, so I thought I would just introduce the country and make sure we're all on the same page as to where it is. So Mozambique is located in southeastern Africa, just north of South Africa and South Tanzania, and I guess opposite Madagascar as well. It's a very long, it's got a coastline, a very long coastline, and we're actually focusing on one specific part of that coastline, which is the Inhambane Province. So that's in southern Mozambique. You can see it up here where my cursor is. And the reason why we focus on this area is because the large filter feeding megafauna that we are studying sort of spatially aggregate in this area. So it's a hotspot for marine megafauna, and in terms of manta rays especially, there are a few sightings sort of north of that region, and it's quite rare to see mantas further north, and then the sightings further south also are quite few and far between. So it's a really good region to be able to see a lot of those animals and also study them on a regular basis. It's quite the Mozambique itself has a long coastline. It's very wild and remote, and the conditions can be quite harsh at times. So strong currents, big swell, often bad visibility, as the divers amongst you would know. But it's an area of quite high productivity, and this is due to some eddy-driven upwellings off the coast, which lead to a sort of increased planktonic biomass on the continental shelf, and that provides a lot of food for our large filter feeders, so whale sharks, manta rays, devil rays, and many more. But not only is it a hotspot for these animals, it is also a hotspot for fisheries. The coastal communities that live along this coastline, their livelihood is predominantly based around fisheries. And a common method that is used in Mozambique is something that's known as gill nets. So you can see in that main photo in the bottom left corner. So these nets are strung out, usually perpendicular to the shoreline, and they can be set from the surface with floats, and sometimes they will go all the way down to the bottom, or they can be set from the bottom. But due to the nature of these nets, they will catch anything that will swim into it, as you can see in a couple of these photographic examples here. So these fisheries pose not only a big threat to mantas, but also to other shark species, sea turtles, and sometimes even cetaceans or dolphins and things that get caught in those nets, when predominantly they're actually targeting smaller reef fish as well. So these fisheries, as I said, pose quite big threats, and it makes the population monitoring and the research that we're doing very important. So the more you know about something, the better that it can be managed and protected. This also means that on the management and conservation side of things, that working together with these fishing communities and sometimes in locally managed fishing areas is essential. In order to make a difference in terms of the conservation things. And also our research is generally designed, as I said, to find out more about these populations so that we can better inform the management or inform the fishing communities as to what we think could be best for the preservation of these animals. All right. And I'm just going to point out here that in Mozambique, we are lucky enough to see 2 of the currently recognized species of manta ray. So the giant or the oceanic manta, which is on your left, and the reef manta ray on your right. And my research predominantly focuses on reef manta rays. So this talk is going to focus on those. So all of the information that I'm going to be telling you today will be about reef mantas, but if you want to ask any questions about giants towards the end, then feel free. And I'm going to focus on 3 topics. The first of them is looking at population trends and demographics. And we did that using the photo identification of reef mantas. So for those of you that have attended an MMF talk or are familiar with manta research, you'll know that the individuals have a unique spot pattern on their ventral surface or on their belly, and we can use that spot pattern to identify each manta ray individually. It stays with them from birth and it doesn't change. So it's a very useful who's who there, and it's very helpful for us for research. So photo ID is generally the kind of backbone of manta ray research, no matter where you are in the world. And the information here I'm going to talk to you about is based on 16 years of data collection in southern Mozambique, so from 2003 through to 2019, which makes it one of the longest studied populations of manta rays across the globe, along with a few others. And it's also based on just over 2,500 hours of time spent by researchers and volunteers doing underwater diver surveys, looking for not only mantas, but also other megafauna. So that's 105 days spent underwater. So we're looking at quite a large and long-running data set. And this work actually carries on from work that co-founder Andrea Marshall did for her PhD. So in 2011, Andrea and the team published a paper here, Size and Structure of a photo ID'd population in southern Mozambique. And that was based on 4 years of data, photo ID data. So the work that we've done for my PhD is now based on 16 years. And so we're just extending on from that. And what we did, because our divers have spent quite a bit of time surveying up and down the coast, we condensed the dive sites into 3 different regions for the different analysis. So you can see on the map here, the top region with the sort of squiggly lines was Bazaruto, so anywhere from Pomen up to the northern boundary of the Bazaruto National Park, which is up here in the orange. And then we split from Pomen, so that's the Bazaruto region. From Pomen down to Pendani, so just south of Tofu, is the Tofu region, and then Pendani just to south of Zavra, we condensed together into the Zavra region. So just looking separately at these regions, we have the total number I've identified individuals below the pie chart there. And then the pie chart shows you the ratio of sort of males to females. So there was 763 individual reef manta rays identified in around the Tofu region, 279 in Bazaruto and 228 in Zavra. And in total across the whole province, we identified just over 1,200 mantas. And then you can see, interestingly, that the sex ratio is quite even in Bazaruto and Zavra, but for the Tofu region, it's a little bit female bias there. And up in the top corner as well, we do have a couple of sites off here, that Ponta de Oro, which is on the very southern border of Mozambique and South Africa, and Sodwana, which is where we've had a few sightings recorded of manta rays down that coast too. But the majority of the research is focused within the province. This gives you an idea of identifications over time. So this is what's known as a discovery curve. You can see here, the stuff in the box on the side is just the dive effort, so I wouldn't pay too much attention to that, but it's just showing you the rate of individual identification over time. So you can see that we did a lot of diving from 2003 to 2009 in the Tofu region, which is this dark turquoise line. And that was the majority of Andrea's work that she did for her PhD, and a little bit further on from that. And then we started diving and had some survey effort in Zavra from 2010 and in Bazaruto from mid-2013. And you can see that those discovery curves are still sort of growing. So that means that we haven't identified all of the individuals in those areas, and there's still a lot of work to be done. And I did want to point out that with the data here, this is all MMF data. We do have collaborators, Nakia and the team at Marine Action Research down in Zavra, who are specifically studying the manta rays just off the coast there, and that's their main study site. And they do have a lot of interesting science that's going to be coming out over the next few years that really focuses specifically on that subsection of the population. So watch this space for that. Something else we looked at was the number of re-sightings per individual for the reef mantas. And the main thing to take from this graph here, you've got number of individuals up the side and then the number of sightings, so the number of times each individual was sighted along the bottom. And what we're looking at here is a very transient population, and this basically means that I think it's 62% of our manta rays have only been seen once. So that's sort of opposing a resident population, which would be characterized by the manta rays coming back over and over again to the study sites and being seen many, many times. We've also got a population, if you look over to the right-hand side, of larger individuals. So this plot here shows the disc width and the number of individuals that fits within that category. So disc width is measured wingtip to wingtip, and it's showing that Mozambique has quite a large population in terms of body size. So we are lacking immature individuals in Mozambique. It's a question that has sort of we've been pondering for quite a while, is to where the juveniles are. So we're seeing a lot of large adults either reaching sexual maturity or already sexual mature. And that is quite different to a lot of different other populations around the world where juveniles are seen almost as often as adults. So there must be some kind of nursery area or area that we either don't survey or definitely don't know about yet, where the juveniles must be hanging out. And then finally, for this chapter of the PhD, I did some photographic mark recapture modeling. I'm not going to go into this too much because it took me months to get my head around. I think it's still actually, I'm trying to get my head around it. But essentially it's mathematical modeling that will use the photographic resites of each individual to calculate the total number of manta rays in a specific area at a specific time. So for us, this area was the dive sites around Tofu Beach. So we've been diving quite consistently there since 2003, all the way through to 2019. So the effort has been stable, and we've also got a number of dive sites within this same region. So I didn't do the photographic mark recapture modeling for Bazaruto or Savra, purely because it's quite a data hungry method. So we focused here on prior to tofu, and I also used the main sighting season of October to March to keep in line with Andrea's previous work. And I know this figure has a lot of lines on here. The thing that you should mainly take into account is this grey shaded area. So this is abundance over time, and you've got the years along the bottom axis there. So we were seeing sort of over 500, well, over 400 individuals in the earlier years. And then this drops down all the way from sort of 800 where it peaks in 2004 and 5, down to after 2013, we were only seeing, it was less than 100 individuals that were estimated to be using that area after those years. So this is quite a drastic decline. It is in line with work that colleagues of mine did previously. So Chris Rona and the team did some modeling from sightings data. So this is like the total number of individuals sighted, or the total number of manta rays sighted on a particular dive, as opposed to looking at individuals as we did with the photo mark recapture. So our work has sort of followed on from what Chris has been doing, and we can see that this decline in sightings has carried on from 2011 all the way to 2019, or basically until now. And you can also see here that there's been a decline in other megafauna as well. So whale sharks, which are in the light grey, And then we've got the giant mantas in the white spots here, but they've stayed relatively stable. And I just wanted to point out that just because we're not seeing these animals, so just because we're not coming across them on our survey dives, it doesn't necessarily mean that they are dead. There could be a few reasons for us not being able to see them. So they could have been landed in fisheries, that's one. They could have been using sites just offshore and outside of our study area, and generally out of our reach as recreational divers. So if the mantas are using cleaning stations sort of at the 40, 50 metre depth range that we don't generally dive and we can't get to, or get to for very long on scuba, then we're not going to be recording those individuals in our surveys. And the other reason is that they could have been, well, they could have moved to a completely different region altogether. And we thought that if they were going to move somewhere like Bazaruto or Zavra nearby, then we would have seen those same individuals and an increase in sightings in those regions corresponding to the decrease in tofu, but that's something that we haven't seen. So potential reasons for why that we've seen this decline. So it could be something that's occurred quite naturally. There is potential that it could have been a shift in habitat use, which would be related to prey availability. And for manta rays, they feed on zooplankton. So an animal that is so large that feeds on such a tiny food source will obviously have to follow that food source to make sure they're getting themselves enough food each day. And so they could have moved, there could be a change in oceanographic conditions that has meant that the mantas have moved into another area, although this is not something that we have evidence for. The other reason is it could be due to human impacts. So fisheries, as I mentioned, there's some small scale artisanal fisheries in Mozambique where around 20 to 50 manta rays are estimated to be caught per year. And so that could have, this could play a part. And then the other thing is that the manta rays could be exhibiting what's known as site avoidance. And this has happened in other parts of the world, generally due to an increase in tourism or dive or boat presence in the area around the particular sites. I just want to note that this photo, the bottom wasn't actually taken in Mozambique. I just used it as an example of dive site overcrowding. And you can see here that there are many, many divers all around this one manta ray. So you can see that this might be something that's quite overwhelming. And this can, this unregulated tourism can sometimes lead to site avoidance. Although I do know a lot of the dive operators in Mozambique do dive quite responsibly and adhere to a code of conduct, but a general increase in a dive tourism industry can potentially lead to manta rays and other animals using other areas just to get away from the crowds. Now moving on to the second topic, we're looking at movements and habitat use. So studying a large, highly mobile animal, especially one that doesn't need to come to the surface to breathe and can sort of swim for thousands of kilometers, it's quite important to know how far they move, where they go, which sites they use, and at what time of the day, and also how these different sites are connected. So the way that we did this was through acoustic telemetry or acoustic tagging. And this is a paper that myself and some wonderful colleagues published just at the start of this year in Marine Ecology Progress Series. So get in touch, I can send you a copy of that if you're interested in reading more about this study. But I'm going to give you a brief overview here anyway of all the good bits. So the way that this works is through attaching a small tag to the dorsal surface of the manta ray, and you can see that in the bottom left-hand photo there. And in the right, you can see one of my colleagues, Dan, who is attaching a tag to a manta ray. So each of these tags emit a unique sound signal every 30 seconds or so, and that's picked up by underwater hydrophones or acoustic receivers, which are placed at different locations along the coastline. And they usually pick up a tagged manta whenever it swims within around 300 meters of a 300-meter radius of the receiver, depending on how noisy the reef is. So believe it or not, reefs can be quite noisy in terms of, well, during the day, maybe boat traffic, but also just general biological noise. So that can affect how far our receivers can pick up those tags. And so what we did was we assembled an acoustic array up and down the coastline. And so in the bottom right-hand corner, you have an example of an acoustic receiver. It's the little black unit here. So we attach those at cleaning stations, which are the turquoise coloured dots and triangles and diamonds here. Feeding sites, and also we put some along potential movement pathways to try and pick up where mantas were moving up and down the coast. Now, we did this in 2 deployment periods. So in 2010 and 11, this focused mainly on the Tofu and Zavra region. And then we expanded that array in 2013 and 14 up to the Bazaruto archipelago and around San Sebastian, to give us a better idea of how the animals were moving up the coastline. We tagged 43 reef mantas overall, and the tags were attached for an average of 118 days. And that ranged quite widely from 13 to around 657 days of the tags staying attached to the mantas. So we got quite a bit of information about where they're moving and how they're using particular sites. So here, these plots are giving you a number of detections versus the time of day. And these are for the different sites at which we place the receivers. So I wouldn't worry about the site names, but if you're just going by the color coding of the box up in the top left corner of each plot. So the dark turquoise sites are cleaning station sites. So these are areas of reef where the manta ray will come in to be cleaned by host cleaner fish. So they take off parasites and dead skin or infected tissue, and this helps with sort of general bodily health, but also with wound healing as well. And what we can see here is that the majority of cleaning station visits occur during the day, with those dark blue shaded areas representing nighttime. So you can see a pattern across all of these cleaning station sites that the mantas were visiting in the day, but they weren't visiting as much at nighttime. There's quite a good quite a good contrast here between the feeding site here. So mid-Bazaruto, you can see that a lot of the detections actually, they began around dusk and then they carried on until dawn, and then there were not many detections during the day. So that's giving us an idea that the manta rays may in fact be using the cleaning station reefs in the daytime and then moving further offshore or to known cleaning sites at nighttime to feed. And then the last 2, the orange sites, were the receivers that we put along movement pathways. So they didn't pick up as many detections. So usually just as the mantas were going past on their way to the different sites. The other thing we looked at is the connectivity between these sites. So these figures here show the sites with the grey circles, and the larger the circle indicates the larger number of detections at those sites. So the larger the circle, the more important that site is to the manta rays, essentially. And then we also, with the different coloured lines, look at the connectivity, so between those sites, so movements from one site to another. And just to let you know that these aren't specific tracks, it's just a representation, otherwise we would have had some of our manta rays swimming over land. But this was the first deployment where we focused solely on Tofu and Zavra. And so you can see that the manta rays are moving between those sites quite well. They're sort of quite an interconnected region. And then when you look at the second deployment, what we can see is that there's also this connectivity here in Prada, Tofu and Zavra, but in Bazaruto as well, between the sites in the archipelago and also in San Sebastian. However, the animals aren't moving, or at least the tagged ones anyway, weren't moving between those 2 location, so weren't moving between the north and the south. So this is giving an indication that the manta rays are giving, showing site preference to an area. Then the last thing we did here with our acoustic tagging data was estimate home ranges for each of the deployment period. So each graph here is one for each deployment, and this is the average sort of core habitat, which is the orange areas there for manta rays, and then the average extent habitat. So the core habitat is where they spend the majority of their time, and then the extent habitat just identifies other areas that they may use. I've also overlaid in the second plot, in the second deployment, the outline of the Bazaruta Archipelago National Park. So within that area, manta rays are protected. They've got fishing regulations on what kind of fishing can occur in that park. So that's offering a bit of protection there. But you can see that the manta rays are using areas outside of the park, and also that the core habitats exist off the coast of Tofu and Zavra as well. So this sort of identifies the priority regions for spatial management and conservation, which come in the form of marine protected areas and such. All right, and moving on to the final topic and technique. The last thing that we looked at was population genomics, and that was looking at the genetic connectivity along the coastline of Mozambique. And how we did this was by collecting tissue samples from 120 different reef manta rays. And the way that we collect a tissue sample is by using a custom-made biopsy tip, which is mounted on the end of a Hawaiian hand sling. So we take a small tissue sample from the individual, usually from the trailing edge around of the pectoral fin, and that gives us about a 1 gram sample of muscle tissue, which we then preserve in ethanol, and then we send it to Australia to the lab. And I actually ended up flying to Mozambique purely to pick up these tissue samples in 2018, because they were absolute precious cargo that we didn't want to trust with couriers. So it was not only a very nice trip, but I also got to go and make sure that the treasure got home safely. So once we brought them back to the lab in Australia, we extracted the DNA. Then we do that by breaking down all of the surrounding proteins and other parts of the cell, so that all that is left in the sample is the DNA itself. And then the DNA is sequenced for each individual, and we identify specific genetic markers, and these are called single nucleotide polymorphisms, or SNPs, which is a lot less of a mouthful. And they are variations in the nucleotides that will occur at a specific point along the genome. And we just investigate the variation in these SNP markers amongst the different individuals. And from that, we can look at things like the genetic structure of the population and connectivity between different regions. We can look at relatedness and kinship of different individuals. And we can also give a rough estimate of the breeding population size, which gives a bit of insight into population health and viability, because obviously the larger the gene pool, the more genetic variation it has, and therefore the greater that population, greater ability that population has to adapt to environmental change. So just to give you an idea of where we collected the samples from, following the other two studies, we got samples from the Bazaruta Archipelago and San Sebastian in the north, and also from a number of different sites around Tofu, from red sands in Zavra, and then one single individual, which was a bit of an opportunistic sample from Port St. John's when Andrea was down doing one of her sardine run trips. And we also compared Mozambican individuals to a small group of individuals that were sampled in Western Australia. And those samples were provided by our collaborators, Amelia Armstrong and her team at Project Manta. So working together with them, we were looking at gene flow across the Indian Ocean just to give a broader idea of population structure. And just jumping back to the results that I was showing you before from the acoustic tagging is that we found that the manta rays in the northern sites were moving quite widely within that area, but not moving further south, and sort of providing connectivity there on an ecological scale with the animals in the south, in Parieto Tofu and Zavra. So our main question here was simply whether we were looking at two different subpopulations. And I should also note that through the photo identification that we could see that there were only 24 of the 1,200 individuals that had been photographed in Tofu and Zavra, but also up in the Basurito archipelago. So we were looking to see whether we had substructuring. So are we looking at two different subpopulations, or are we looking at a single breeding population? And it turns out that what we're looking at is one single breeding population. So this plot here just gives you an idea of all of the different individuals. Each dot is a manta ray, and the color denotes where that manta ray was sampled. And so seeing as these aren't forming distinct clusters, they're sort of all hanging out together as one, this indicates that we're looking at a single breeding population, and the little South African individual is also sitting within the Mozambican population there, although there's only one, so obviously we need to have a better look into that with more samples. And we also estimated the breeding population size of around 400 individuals. And I should note that that is the representation of the generation before the one that we sampled, so the parent generation there. So for manta rays, they've got quite low reproductive rates, they've got long generation times, so we're looking at a snapshot into the past for that. So that's giving us a general idea of where the population is at. And if you're thinking that doesn't sound like many manta rays, well, it's not really that many manta rays. So it's also something we need to consider when implementing some management and conservation. Then looking across the Indian Ocean, you can see that Western Australia, which are these dark grey dots over here, and the Mozambican population here are completely separate. So this is indicating that it's just not much gene flow across that, across the Indian Ocean. So these are two completely separate breeding populations. All right, and just to finish up, I just wanted to give you an idea of the kind of conservation applications of the information that we've discovered through that research. As I said, that we mainly try to gather this information about each different species, so we're able to better guide management and better guide protection. But just so to give you an idea of where we're at with that, we identified the declining population trend that is carried on from the previous studies in tofu, and we've also provided baseline numbers for the other regions for Zavra and Basurito. So that also gives, like, identifying that trend gives a bit of a sense of urgency for the management of the Mozambican population. It's a long and well-studied population, but it is also one that has been used many a time as an example for a population at risk. So it's quite clear that we do need to start putting some management strategies into place, otherwise we could be looking at a localized extinction if things continue the way that we're going. Through the tagging work, we identified important habitats, so those are areas that should be prioritized for conservation, and that's in terms of things like marine protected areas, whether that be the locally managed protected areas or areas that fishing regulations are put in place. We've sort of provided that information, and we've identified that it's a single breeding population, which means that any management needs to be put in place across the whole coastline, and even having that preliminary information about the South African manta ray that fit in there with the same population may give an indication that we actually need to coordinate management efforts internationally, so between South Africa and Mozambique as well. So this is just the first step really here. As scientists, we have provided that information, and now it's time to work with management bodies, whether it be nationally or locally, sort of locally managed national parks, and also working with local communities to be able to start to put some management and protection strategies in place for this population. And I just wanted to end by just giving a special thanks to not only the lovely people pictured on screen, so my colleagues at MMF, and also my supervisory team, but also all of our supporters in Mozambique, and to the funders, and also their Megafauna Foundation volunteers, everyone that put their time into diving and data collection and management as well. And thank you also to you guys for listening. And I'll pass it over to Janae now so that we can answer some questions.
Great. Thank you so much.
No worries.
All right, we got a couple questions so far down in the chat. And then if anyone who tuned in maybe had a question but forgot to enter it in the chat, have at it whenever you'd like. So let's see what we have. Okay. Chris says that it looks like the discovery curve was pretty flat for tofu recently. And if you know if there's new mantas that come into the population that are juveniles, small little mantas.
So the discovery curve for tofu is pretty flat because we're really just not seeing that many animals there. I think around 2017 and 2018, there was like 3 and 6 reef mantas that were sighted. So it may not be that we've discovered all of the individuals there or identified all of the individuals there. It's just that we're just having so few sightings that it's starting to flatten off. But we're still not seeing many juvenile mantas around at all. It would be nice because that would mean there's new recruitment into the population, but it isn't what we're seeing. A lot of the recently identified individuals are still mature adults.
And then I saw in the beginning when you were showing your sites, and that tofu for the reef mantas had 763 and the other sites had less than 300. And that it was just like drastically more mantas there in that particular area.
That's also because of research effort. So we've been working a lot more in tofu for a lot longer, and also we've got consistent dive effort throughout the year. So with the discovery curves for Zavra and Bazaruto, they were still rising. And I know Nakia and her team have identified more than 200 individuals that visit Zavra every year. So that is also because just of purely of research effort. And Andrea, and supposedly me, when I get out there, will be focusing more on the Bazaruto region. So we will obviously be building up the number of individuals there as well as we keep seeing more new mantas.
On your chart that you had the different cleaning stations and the activity throughout the day and how it spiked in the afternoon and fell off in the evenings, and then you had the feeding station, which was like the opposite. Chris asks, how did you know that that was a feeding station?
Oh, it had been identified through aerial surveys. So that was actually daytime feeding that we saw of large aggregations. And so we just put the receiver there to see if we were going to pick up lots and lots of mantas at particular days of the year when the plankton all gathers in that area. But what we actually found was that we were seeing more detections during the nighttime. So spending the time there at night is more of an assumption that they were feeding there at night, but that's based on us seeing them feeding there during the day. It's quite a shallow, sandy bottom, and there's no sort of reef there that would be habitat for cleaner fish, so it's certainly not an area that they would use as a cleaning station. they could be passing by, but it's unlikely that we're seeing that many detections of the mantas in that area for one time if they're just sort of zooming past.
Then speaking of areas that they might be attracted to, when you were doing the graphs with the circles and the connectivity, and the bigger the circle, the more you saw in GC, Giant's Castle maybe? It was such a huge circle.
Yeah, so that indicates that mantas are spending a lot of time at that site. So obviously if the tag is pinging every 30 seconds or so, if a manta ray spends a few hours at that cleaning station, then that's going to increase the number of detections at that site. So we're seeing quite a lot of individuals there, but we're also seeing them stay there. And I think the longest time we had recorded at a cleaning station during that study was just over 7.5 hours of continuous detections.
It's a long chill time.
It is a long chill time.
Is there anything noticeably different between that specific site and the other ones surrounding it that you noticed?
Not necessarily, but it could be something to do with like the density of cleaner fish. So if you're a manta ray and you wanted to be very efficient, you would visit a site that has a lot of cleaner fish, and they're obviously going to be able to take more dead skin or clean up more parasites from you than if you're just visiting a site that only has a few. So yeah, we see a lot of site preference of in different places around the world where mantas will come back to that very same site. In Raja Ampat, a colleague of mine looked at whether or not this could be related to social preferences. So the same as we have our own local pub that we like to go to, or our local coffee shop, they know our name. Maybe manta rays have sort of sites that they like to visit over, like preferably over other ones.
Yeah, sure. Like they get treated really well by this particular cleaner fish. They're gonna go back.
Yeah. Go see Larry. He knows what's up.
Yeah, tell him I sent you. We got a question just to kind of go over the tissue sampling again. So this question just says, were you collecting dead skin off the rays like you would a cheek swab, or are you physically removing a little bit?
No, so we're physically removing a bit of tissue. It's probably the equivalent of, I guess, like us taking a fingernail clipping. It's quite a small piece of muscle tissue. Probably the tip would go about 1 inch or so into the manta ray. Now, fish, sharks, and rays, they process pain differently than we do. So often you don't really get a behavioral response at all. Sometimes the manta will get a little bit of a shock and swim off, but we usually see them coming back and cleaning on that same reef or in that same area. And sometimes you just go absolutely no response at all. And as they're quite good at healing, usually within 2 or 3 days, you can't even actually see the site where you took the tissue. And we have actually trialed using the mucus, so coming up and sneaking behind the manta ray with a toothbrush and then just rubbing a little bit of that mucus and trying to extract DNA from that, which is definitely like a less invasive method. But unfortunately, for the SNP markers that I used and the next generation sequencing, you don't get a high enough concentration of DNA to be able to sequence those markers and as much of them as we did. So what we usually do with those samples as well is we split them up, and they're used for genetics, but they're also used for stable isotope and fatty acid studies. So causing that little bit of disturbance to the animal, but we're using that tissue to gain quite a lot of information about genetics and about their feeding ecology as well.
Yeah, that's cool. I've seen a video of when Andrea did a tissue sample, and you would think like if someone kind of took a hole punch of your skin, it might bother you, but the manta was completely unbothered. It was amazing.
Yeah, I mean, some of them get a shock and they don't like it, and they will sort of zoom off into the distance, which is understandable, but some of them you have absolutely no response at all. So maybe they have different pain thresholds like we do, or maybe some of them are just chillers.
They didn't know you were going to do that. See what we got. Okay. David asks, on one slide you showed the home ranges and that they seemed rather small, and then you also mentioned the breeding populations spanning the sites. Do you have estimations of total ranges that they would travel over a lifetime?
Yeah, so the reason that the home ranges looked quite small as well is that they were restricted to the area covered by our acoustic array. So that kind of analysis can also be used on satellite tags. And because that tag will give the location of the manta ray like outside of that specific area, then you can get like a better idea of home range. But the manta rays have been recorded traveling, I think it was 1,100 kilometers was the largest point-to-point movement, and that was along the east coast of Australia. And that was through photo identification. So that's the longest recorded point-to-point distance of manta rays moving, but they obviously can cover quite a bit of ground if they need to. So it's quite likely that they are traveling, they're able to travel up and down that coastline. I think the distance between Bazaruto and Zavra is around 250 kilometers or so. So it's well within their movement range, and also means that they'll be capable of traveling further south, down into South Africa as well.
The genetic dot where you had that random South African ray and to show that they were still part of the same like subpopulation was really cool to think that you have theoretically this range from Bazaruto all the way down to South Africa. Do you know if there's going to be any further like efforts to get more data from those opportunistic mantas that you might find down there?
Yeah, for sure. We've actually got Michelle Carpenter, who is doing she's just changed her master's to a PhD at the University of Cape Town. And she, at the moment, is in South Africa. And she's looking to try and collect some more tissue samples for us, and be working together with her to sort of expand that there. But also she's ramping up the photo identification efforts to try and see if we can detect any movements between Mozambique and South Africa through photo ID as well. The thing with South Africa is that up until now, the sort of sightings have been quite opportunistic, and every now and again we get a diver or an operator sending in photos or a video that might have been taken. But now that we have someone like rallying the troops down there, then I think that we'll be able to get a bit more information and get a better idea of how the mantas are using that coastline as well.
Yeah, that'll be super cool. Let's see, we got the large number of single sightings of individual mantas suggests a lot of movement between the sites. The acoustic study suggests that movement isn't between the sites. Is it the assumption that the movement is to deeper offshore sites or to entirely different regions?
It could be either. The assumption, I guess, if I understand the question correctly, is that they could use the deeper regions to move between those sites, and therefore they weren't picked up by our acoustic receivers, which were located in the shallower areas, because we put them down and we have to pick them up again every 3 to 6 months. So it needs to be in an area that's quite accessible. And for us, we were scuba diving to maintain the array. So that needed to be sort of less than 40 meters or so. But I'm just rereading the question.
I butchered it when I was trying to read it to you as well.
Acoustic studies suggest movement isn't between the sites you studied. Well, what it could be is that there's just a small amount of movement as well. So it doesn't take many breeding events for genes to sort of be passed on and maintained through the gene pool. So it could just be that there are only a few individuals that are meeting up offshore and maybe mating, but that's enough to maintain a homogeneous population.
I have another question for you. I know you are an expert on Mozambican mantas, and my question brings us over to Hawaii a little bit, so I'm just kind of picking your brain on your thought. I was lucky a couple of years ago to have the chance to go to Hawaii, and while I was there, they do this dive with manta rays at night where really I was just holding on to the surfboard face down in the water at night with the lights shining down so the mantas could come.
I've seen so many photos of this.
It's really cool, and you don't touch them. They come and feed. It's very just like watch what happens and be in the magic. But I was wondering if you had any opinions, because we were talking a little bit during your presentation about kind of like maybe mantas would move away from sites as they become more populated. And I didn't know if there was even any data on like the manta populations, because it seems very important to like tourism and to bring people in to that population in Hawaii. And they told me it's residential and they stay here all the time. If you have any idea if the population there has decreased as the tourism has increased, or if maybe those mantas didn't care as much?
Yeah, so I've never been to Hawaii myself, and I haven't worked on that population. So, but I do, my opinion on it, I guess, is that the lights that they use, the dive lights and the big spotlights underwater, attract zooplankton, and therefore the mantas will come to feed on that zooplankton. So it's kind of like a little bit of an attractant. In Mozambique, we don't actually see mantas feeding as often as we do cleaning. It's quite rare that we will come across mantas just sort of like surface feeding or anything like that. So when they're feeding, manta rays are sort of very focused on what they're doing. They've got their eyes on the prize. And you probably saw that they would come like really close to you if they've got impeccable spatial sort of sense, and just carry on what it is that they're doing. So they're less likely to be disturbed while they're feeding. There is sort of some discussion about whether or not that's termed as provisioning in the same way as feeding sharks or chumming for sharks or something like that could affect the natural behavior of the sharks that may become sort of reliant on that feeding. But I just think the amount of food that manta rays have to eat like every single day, and also the fact that I think, correct me if I'm wrong, anyone, but that they were feeding at that site before they started putting the lights up anyway. So the lights are just going to start concentrating that plankton together into a certain area, which means that people can view it there. So I don't think that they've seen any changes in the abundance. It would be a really interesting study because that's a really unique tourism operation out there. I don't think there's anywhere else in the world that does it to that scale. I know some dive boats in the Maldives do it just by chucking a spotlight off the back of the boat, and then it attracts mantas and sometimes whale sharks and things. But in Hawaii, it's like this massive operation, isn't it? So yeah, I don't think they've seen a drastic decline, but I would be super interested to talk to the guys that work out there and get their opinion on that.
Yeah, cool. And kind of like, oh, they're there to eat, so they're super zoned in and focused on that buffet. But if you want to go to the spa, you want to be left alone, and you're going to find a new spa if it's surrounded by us disgusting humans, I guess.
Exactly. And also mantas, when they visit the cleaning stations, they'll slow down, they will hover, they will open up their cephalic fins, and so they're leaving themselves quite vulnerable to things like predation, and also so they are a little more skittish at cleaning stations, and I know that it's most parts of the world, the divers will try and stay outside of the cleaning station and give the manta a bit of space. But if you've ever been in the middle of a group of feeding mantas, you'll know that they're not bothered that you're there. They'll come as close as they need to get the delicious plankton, and then they'll move on to wherever they need to be. So that's definitely an advantage that they have out there in Hawaii.
And then in the cleaning station lights, are there any studies that you know of that are kind of correlating, I guess, coral health to manta decline? I imagine that'd be pretty important.
Yeah, definitely. I don't think that there are any studies published, but I know that there's definitely been some sort of master's projects looking at that, but I don't know of any that have done it in sort of great detail. But the problem with that is you're going to be wanting to look at declines over time. So sometimes that involves very long-term data sets. So if you want to look at like the state of the reefs 10 years ago, then you have to have surveyed the reefs 10 years ago. And I know, and I have had discussions with Andrea that that for her, she's wishing that we had that data from Mozambique, because maybe then we could correlate like a lack of coral cover or a decline in fish numbers. And then that could be something that is the reason why we're seeing less manta rays, but we don't have that data. So it's something that I think Michelle as well, that I was talking about, who's working in South Africa, is wanting to look at.
Michelle, she's doing it all.
She's great. She's got a lot on her plate over the next few years. But I think that was something that she was wanting to start up, at least collecting that data to make it a long-term monitoring thing. And also I know that the BRUV program in Tofu is collecting that sort of information. I'm not quite sure, you probably know more about it than I do, whether the BRUVs are being dropped at cleaning station sites or whether that's being included, but that's only been started in the last sort of maybe 2 or 3 years, I think. So we don't have that long-term data either.
10 years from now, again, we'll have those answers.
Mm-hmm.
That's all I got. We got some good questions. Thank you so much again for your time to come and present all your knowledge to us, and thanks everybody else for tuning in.
Yeah, thanks guys, it's been great.
Yeah. If you guys are around, we're going to try to do the same type of thing next Friday. So just wherever you found out about this talk, we'll try to post again for our next guest speaker. But thanks again, Steph. It was super great hearing what you do.
Awesome. Thanks so much. See you guys.
Bye, everybody.